Organizers. So the second half of thinking about regionalization and organizers. Now, the initial regionalization of the AP and DV axes plus the germ layer determination, that's going to involve those anterior and posterior organizers, as well as things like the Spemann-Mangold organizer. But then we also have organizers that are specifying major body parts in specific regions, so like a limb organizer. So what can organizers do? Well, they can set up body axes, they can set up body part axes, and that's what we're going to look at in just 1 second. And they can also specify a tissue as a specific body part. For example, PAC6 specifying eyes, and that includes serially repeated structures. And so these latter two can be affected by Hox genes and their effectors. So we already talked about head versus tail organizers and AP axis organizers, and this is the limb organizer. This is the tetrapod limb organizer. Tetrapods are our four-limbed vertebrates. These are mostly our land vertebrates, though of course they also include land vertebrates that went back into the ocean, like whales and otters and seals. So this is the whole limb organizer, is this whole mess here. And when people talk about the specific limb organizer, sometimes they're only talking about this chunk here, and sometimes they're talking about the whole thing. So just keep that in mind, that people will sometimes only be talking about this little piece here. So there are two parts of the whole limb organizer for looking at the more broad view of it. We have this part in red here called the apical ectodermal ridge, or the AER. And this is going to just be the distal-most tip of the limb. It's going to have this ectodermal covering that secretes FGFs down onto the mesoderm that's underneath. So underneath, this is like all mesoderm down here. So these FGF signals, these are diffusible morphogens. They're proteins that can diffuse, and these are ligands, like we talked about last time. These ligands can bind to receptors. When these ligands find a cell with a receptor on them, especially a mesodermal cell, they tell that cell to grow like crazy. So this signal from here causes cell proliferation underneath it. And so if you basically have like a little growth zone here and tissue behind it, and it's telling all this tissue to grow, you get growth in this sort of a pattern. And we're going to talk about that more later on in the semester, about growth patterning and Turing patterns. So the ZPA, zone of polarizing activity, this is a group of cells that secrete sonic hedgehog. And sonic hedgehog is another ligand. It's a hedgehog brand ligand. And this is also a diffusible morphogen. So this sonic hedgehog diffuses through the extracellular matrix, and it's hitting all of these mesodermal cells around here that have hedgehog receptors on them. And when these hedgehog proteins bind to the hedgehog receptors, that causes a signal transduction cascade. And depending on how much sonic hedgehog you're exposed to, you're either going to become pinky side, that's if you're exposed to a lot of sonic hedgehog, middle fingers, that's if you're exposed to medium levels of sonic hedgehog, or thumb side, that's if you're exposed to absolutely no sonic hedgehog. So this is anterior to posterior axis determined by this gradient of sonic hedgehog. So the proximal distal axis itself, that's this axis here, this actually gets patterned through timed expression of different Hox genes. I'll talk about this a little bit more later in the semester, but there are all of these like really neat features of this. The way that this expression pattern happens, it's through actual developmental timing, not just induction from cell to cell. That's involved too. It's kind of amazing. And I want to talk about it now, but I actually need to finish up organizers. All right, so another type of organizer. This is the kind that specifies a serially homologous structure. So for example, the same organizer, it's used to specify your arms and legs, but the downstream genes really differ depending on if they're attached to your shoulder girdle or if they're attached to your pelvic girdle. So likewise, lobsters and shrimp and other decapod crustaceans, they have the deployment of the limb organizer on most of their segments. And what that limb organizer actually produces is going to be dependent on what Hox genes were expressed in that particular segment. We're going to talk about this more a little bit later with insect wings. But first, I want to kind of show you, this is putting serial homology plus these organizers together, plus the Hox gene. So kind of everything all together. So this is looking at how regionalization acts on top of these repeated structures. So here, these two are fruit fly here. And so we're going to start with those. So here we can see this wing organizer, and this wing organizer is going to be expressing vestigial and scalloped. So these two genes, vestigial and scalloped, are the wing organizer genes. Now here we have wing program active, but UBX, this is ultrabithorax, it's a Hox gene, and this is expressed in thoracic segment 3 plus in the abdomen. And this actually turns off a lot of these wing programs. So if this UBX is around, that either converts this wing program into nothing, it shuts it down entirely, or in segment T3, it converts it into the wing into a haltere. So a haltere looks like a little tiny mini wing, but it's actually involved in proprioception. So likewise, the limb organizer in Drosophila normally expresses distalis, homothorax, which I always think is hope that helps, but it's not hope that helps, it's homothorax, and extradenical, EXD. So distalis, homothorax, and extradenical. That's the limb organizer. So you can see it's expressed here, and then it's also expressed in the leg, distalis, homothorax, and extradenical. And what makes the decision of antenna versus leg is the Hox genes that are present. Do you have antennapedia, sex combs reduced, and ultrabithorax? These are all these trunk genes. So if you have overexpression of antennapedia, that's just one of these trunks Hox genes, you have overexpression of it in the head, you convert this cassette here into a leg. And that's how you get these antennapedia mutants with the legs growing out of the top of their head. That's just converting this normal limb organizer that's normally expressed in these antenna regions. It's just converting that into leg instead of antenna. So a choice between these serially homologous structures. This one here is moving away from Drosophila for one. So this is looking at vertebrate limb organizers. And like I mentioned before, they have to make the decision between forelimb and hindlimb. And they do this based on the expression of TBX5, which is expressed up in this region of the body, versus TBX4, which is expressed down in the pelvic regions of the body. And so it's the same limb organizer, but it's going to be changed based on TBX4 versus TBX5 expression. So like I have promised you, and I know you're so excited about this, we're going to do a deep dive on the Mangold Spemann organizer this week. So here's just a little quick reminder of Xenopus gastrulation. So here's our blastula. We get involution around the lip of the blastopore, so around sort of this like equatorial ring around the embryo. And the involution is going to start at the dorsalmost point of that, the dorsal lip of the blastopore. Meanwhile, we get epiboly happening of these ectodermal cells to cover the whole embryo. So then we have this postgastrula that looks like this, where if you are ectodermal cells that are getting signals from this notochord here, you're going to get the signal to turn into CNS. If you're away from that, you're going to get the signal to turn into skin. So really quickly, we know a lot about this because of these cortical rotation experiments where we take this gray crescent, these cells that used to be in the gray crescent region or that developed from gray crescent region cells, and you slap them on the back of a different embryo and you get a twin tadpole. If you get rid of this gray crescent by preventing cortical rotation, you get a belly piece. This is just skin and endoderm, no mesoderm. So what specifies the organizer, and how does the organizer signal to surrounding tissues? Well, it's both complicated and simple at the same time. So we're going to look at a slightly pared down version of this. There are other genes involved in this, but I'm just going to focus on sort of the major players in it that have really big effects. So first off, let's start with just going back to our egg that's just about to get fertilized. So here in the animal pole, we have these genes called VEG-T and VEG1. That veg for vegetal, that are in the animal pole, as well as disheveled. So disheveled, this is that green blotch that we've been seeing. That's actually going to be disheveled that's tagged to the cell membranes. All right, so VEG-T and VEG1 are going to turn on these two genes, BMP and nodal. And the version of BMP that we're going to be talking about here is a TGF-beta homolog. And also, it's going to turn on these endoderm genes, which I know sounds really mean, but like I have a hard time really caring about the endoderm. I like never think about it. So endoderm, like blah, sure, it gets made, I suppose. If anyone's interested in endoderm, we can definitely cover it, and I'm willing to learn more. And I feel a little bit bad that I neglect it so often. All right, so after cortical rotation, so that's just after fertilization, this big blotch of disheveled gets moved over closer to these anterior regions. Okay, so now disheveled is actually going to inhibit GSK3, which we saw a million years ago as part of this Wnt pathway. And GSK3 is going to have, it always has the effect of inhibiting beta-catenin from moving to the nucleus. And beta-catenin would normally turn on nodal. So if we have disheveled around blocking GSK3, that means that now we get the activation of beta-catenin. We've turned off the beta-catenin repressor. And so now we're able to make nodal in all of these regions. So now we've got this sort of like nodal gradient here. So we have this beta-catenin gradient like this, and we have this other nodal gradient here. And that's because nodal, remember, is also affected by VEG-T and VEG-1. So that means we get the highest amounts of nodal where we have this sort of point source of beta-catenin, and then it's sort of graded based on whether or not there's, it's graded in this direction based on whether or not we have VEG-T and VEG-1 around. So high levels of nodal where we have VEG-T, VEG-1, and high levels of beta-catenin. And so that gives us this like interesting gradient of nodal in this direction here. It's like a sort of double gradient. All right, so these vegetal pole genes, VEG-T and VEG-1, turn on BMP and nodal. Disheveled blocks GSK3, releasing beta-catenin, which turns on nodal in adjacent cells. And then this nodal converts the adjacent nodal minus cells to mesoderm. So nodal itself won't act on other nodal expressing cells. It turns the next door cells into mesoderm. And that's how we get this stripe of mesoderm. And the mesodermal marker here is called brachyury. So brachyury is just a gene that's only made in mesoderm, so we call it a mesodermal marker. Okay, so that's converting these nodal minus cells into mesoderm. So this is going to basically be this like stripe layer right on top of these endodermal cells, which are expressing nodal in this graded pattern. And so nodal is now expressed in this gradient just in the endoderm with a stripe of brachyury expressing mesoderm right on top of it. And cordon and noggin are going to repress BMP. And so cordon and noggin get activated in these cells that will become the future notochord. So just in this one little patch here where we also have high levels of beta-catenin, that's where we're going to get cordon, which specifies notochord, and noggin. And cordon and noggin together are going to repress and be repressed by BMP, and BMP itself is going to repress the formation of the neuroectoderm. So basically you don't get conversion of the ectoderm into nervous system if you have BMP around. And so cordon is going to be blocking this. So that means that where you have cordon, the next door ectoderm can be competent to become neuroectoderm. So here's looking a little bit more at these genetics. So that's like kind of thinking about our germ layer specification. But as the organizer involutes, it's going to be signaling to the ectoderm above it to become the neural tube. And the organizer itself has AP polarity because it involutes in an order. So as it involutes, the leading edge, those are those first cells that involute and move over to the anterior side of the embryo. They express something called Cerberus. And the lagging edge, which is going to be the posterior end of the notochord, they express FGFs. And so we end up with sort of like this graded gene expression along the AP axis of the notochord, where at the anterior end, we get forebrain forming, and the marker for that is OTX. In the midbrain regions, we're going to have engrailed, this is OTX is in the overlying ectoderm, excuse me. In the midbrain regions, the overlying ectoderm here, that's going to be engrailed. In the hindbrain, we're going to have things like CROX9. So again, in the overlying ectoderm, just over this part of the notochord. And then in the trunk tail regions, the neuroectoderm is going to express HOX6 through 13. So the notochord patterns these anterior posterior fates, and it also organizes the important difference between skin and nervous system fates. And then later on in development, it's going to actually help to create the dorsoventral axis of the neural tube itself. And that's what we see here. And this is a common process to all vertebrates. It doesn't really matter how you gastrulate. Once you have this notochord established, you get organization of the neural tube specified by the notochord and the overlying epidermal cells. So now instead of looking at the embryo like this, we've switched like this and sliced right through it. So we're looking down at the sort of the neural tube, which is going to become the spinal cord this direction, and right underneath it is the notochord. So where you have the notochord around, the notochord is secreting sonic hedgehog again. And if you're neural tissue and you receive high levels of sonic hedgehog signal, you become motor neurons. If you are neural tube tissue, neural cells, and you receive high levels of these TGF-betas or bone morphogenic proteins secreted from the epidermis, you become sensory input neurons. And if you're in between, you become interneurons. Okay, so those are our organizers, a couple of important organizers, and we'll look at some more examples of these in the future. If there are any organizers that you're interested in, let me know, and we'll see if we can cover those towards the end of the semester.